Okay, hello everyone, welcome back to our YouTube channel AgriAddict. So as you people requested for the ASRB most expected question series for the seeds and the technology along with the genetics and plant breeding. If you know, like we have already done it for the genetics and plant breeding in our online course of the mock test bundle for the ASRBNET. Similarly, students of seed science also requested that we also want this most expected series for the ASRBNET exam 2025, which is most expected, and it will be mostly held on the dates that are mentioned by the ASRB. So if you know that, like there were rumors that the exam will be postponed, but mostly the exam will not be postponed. It will be between like 3 to 5. So dates and the hall ticket will be coming soon. So if you are preparing for this, so this is like from 2 to 6 of September. So this will be mostly like from this week or next week, like the hall ticket will also come. The exams centers will be also clear. So if you are preparing for these exams in the subject of seed science, so please prepare for the exams, and the cutoff and everything is not too much high. So if you prepare with the MSc knowledge that you have, you can definitely crack these exams. And to help you people, like we are coming up with this like most expected series, plus we also have like complete mock test bundle. See, complete mock test bundle is available with us, so which will help you to practice the questions. And mostly like we have like 30 plus mock tests which are available in this course, so in which like most of the questions related to the seed science is already clear. See, if you attend those questions once and get to know the answers from those papers, where like explanations, most importantly, like we have also added the explanation for each and every questions. So if you just try to attend it once or twice, many things will be clear. Okay, in this last moments, if we like better to just attend the mock test as many as possible, so that most of the questions will definitely come from the test that you are going to attend, right? So for this, the attending the mock test is very, very important. So all the questions that we have given in our mock test, those questions we'll be discussing here, so that everything will be clear. Wherever you have doubts, you can comment on this video, and if you are a student of already a mock test bundle and the master course, like you can directly post it on the master group of WhatsApp. There we will assist you with all the doubts that you have, right? So as per your requirement and the request, we have finally come up with this ASRBNet series, where we'll be discussing the important questions, not only just the question and answer, but also the related concepts. Today, to discuss these things, we have one of my junior from the seed science and technology, who have recently completed his MSc, and he has given his PhD exams, and he has secured really well in the exam, and he will be mostly expecting to get All India 3 rank, and he'll be coming to the PhD at IRA again. And he has like thoroughly prepared for his MSc, where he has sufficient knowledge, where he will be sharing with all of you. So if you have any doubts related to this, you can just either drop in the chat box, or if you are watching this video after the live, you can comment on this video, and we will definitely try our best to like attend and answer all the questions that you ask. Manja, this is yours, and please introduce yourself and start this.
Thank you, sir. Hello everyone, welcome back to our YouTube channel AgriAddict. Myself Manja Sir, currently pursuing MSc in Division of Seed Science Technology, and I given PhD SRF examination in the Division of Seed Science Technology. I got marks of 306 in the ICAR PhD exam last year, and today I cleared CSRNet in the December series 2024. Today I am here to discuss the expected question for the ASRBNet SMS STO for the 2025. Here, before going to for the questions, I will explain the exam pattern of the ASRBNet SMS STO. For in exam there is a 150 question will be there. In that, for each correct answer there will be one. You will get 1 marks. If they made wrong, if they incorrect, there will be negative marking of 0.33. So the minimum qualifying marks for getting net is for general category, it will be 75 marks, that is 50%. For EWS and OBC, the minimum qualifying is 67.5, that is 45%. For SC, ST, PWD category people, the minimum qualifying marks is 60%. 60 marks, that is 40%. Let's start our discussion. Is the first question. the coordinate of the monocot is known as. Before going to discuss each question, I would like to ask you a question. We will directly discuss a question, or else you have to give some explanation for some topic, and we will go discussion. And before, you know that seed technology will start with the word seed. What is the meaning of seed? Seed is a fertilized ovule which consists of embryonic axis and reserve food material. We know there is a monocot and dicot. The cotyledon is called as scutellum. Is cotyledon of monocot is called as scutellum. What is the cotyledon of dicot? It will be cotyledon only. The region of water absorption in seed. The second question, the region of water absorption in seed. Here option is micropyle, hilum, aerials, rapid. Then for seed has to germinate, it has to absorb water and it will radicle will protrude after imbibition process. For absorbing the water, which part is helpful? For example, in seed, the part is called micropyle, where the seed will absorb the moisture. You can know that we have know the embryo sac part, that is called as female gametophyte. I will
Recording in progress. Recording stopped.
Let's assume this is the female gametophyte. Here, you know that in female gametophyte, the ovule is surrounded by integuments. And the center portion, this is called as central nuclei, or else polar nuclei, or else also called as secondary nuclei. The portion of the embryo in embryo sac, the portion where integument is not covered is called as micropylar region. The portion which opposite to the micropylar region is called as chalazal region. I am here discussing the parts of the embryo sac. Towards the micropylar region, there is a 3 cell, where one is egg cell and 2 are antipoda. The 3 cell towards the chalazal region, they are called as antipodals. Sorry, the parts 2 cells, that is other than the egg cell, they are called as synergid. The cells towards the chalazal, they are called as antipodals. The cells towards the micropylar, they are called as synergid, and there will be one egg cell along with the synergid. This is about the structure of the micropylar. For seed to absorb the water, the portion is to micropylar, because there is a pore is there. Already the integument is not covered by the seed. So this is a micropylar. Answer is micropylar. You know, nucellus, third question is, nucellus substantially contributes to the storage tissue, has perisperm in. You know that after the, I will explain the embryogenesis process and the microsporogenesis process first, and we will going to discuss the questions afterwards. First, for any development, for seed development, first there is a gametes fusion will be there. There is a sexual reproduction will be there. For sexual reproduction, the gamete production. First we will discuss the microsporogenesis and megasporogenesis. Microsporogenesis, The formation of microspore from the megaspore mother cell is called as microsporogenesis. The formation of megaspore, that is female gametophyte or the embryo sac, is called as megasporogenesis. For the development of microspore, that is pollen. For pollen development, first microspore mother cell will undergo meiosis division. For any gamete production, the cell division will be meiosis. This microspore mother cell will undergo meiosis to produce 4 microspore, and these will give the pollens. This is a process. But in megasporogenesis, the megaspore mother cell will divide by meiotically to produce 4 megaspore. In that, it will going to in these 4, 3 will degenerate. Three will degenerate and one will go mitotic division to form a embryo sac. I already told you the structure of the embryo sac. The embryo sac development by like first 2 cell, then 4 cell, 8 cell, then going to form embryo sac. But I have missed one point. You have to remember the thing in embryo sac. For example, how many cells are there? Can anyone answer what is how many cells are there in the embryo sacs? Embryo sacs is the 7-celled 8 nucleate stage. You can able to see that 2 synergid, 1 egg cell, 3 antipodals, 6 and 1 central nucleus. This is 7 cell. How 8 nucleate? But the genetic constitution of the central nuclei will be 2n. So it will be 8 nucleate stage. This is an important question. You have to remember this. This is about microsporogenesis, megasporogenesis. Then, after the gamete production is over, the gamete production, in microspore will fuse. Microspore will go into mitotic division to form 2 cell. This is I am going to explaining the pollen gamete formation. This microspore is going by mitotic division to form 2 cell, that is vegetative cell and generative nucleus. This another mitotic division to form by the generative nucleus will go divide into 2 sperm nucleus. The vegetative cell, what vegetative cell will do, what sperm nucleus will do. Vegetative nuclei will going to form pollen tube, but that sperm nuclei will going to fuse with the egg cell and the central nuclei. If from the microspore there will be production of 2 gametes, that 2 pollen grains. That one pollen grain, that one sperm nuclei will fuses with the egg cell to form an embryo, and another microspore like the pollen grain will fuses with the central nuclei, will fuse to form an endosperm. Is also called as primary endosperm nuclei. The genetic constitution of the primary endosperm nuclei will be 3n. This is about the endosperm development. Here question is nucellus substantially contributes to the storage tissue as a perisperm in. You know and me know that endosperm is 3N, but here has been perisperm. It is also a storage tissue where it was produced directly from the nucellus tissue. What is the genetic constitution of perisperm? Before going to this question, I will ask you some questions like what is the genetic constitution of perisperm? The genetic constitution of perisperm will be 2N because it was directly developed from the nucellus cell. Then, in which crop the perisperm is a storage tissue? In sugar beet, it is the storage tissue. What is the function of the endosperm? Endosperm will provide the food material for the growth of the embryo. But in monocot seeds, they are endospermous. Then in dicot seed, they are not endospermous. Because during the development of the seed, the embryo has consumed the food from the endosperm, so endosperm degenerate. But in the monocot seed, it is there. So endosperm, in another name of the endosperm seeds are also called as albuminous seeds. And for non-endosperm seeds is also called as non-exalbuminous seeds. You have to know the example for each category. Like they will give the which among the following is the endospermous seeds, which one of the following is the albuminous seed, which among the following is the exalbuminous seeds. You have to know these points. Which one is the reproductive unit having the embryo, reserve food and protective covering? I told you the definition of seed. Here the definition of seed is they given. Seed is a fertilized ovule. It consists of embryonic axis. Which are the embryonic axes? That is radical, plumule. These are the embryonic axis of the seed. Fruit. How fruit will develop? Fruit will develop from the ovary. Next question. The microgametophyte is also known as. I already told what is megagametophyte, female gametophyte, embryo sac. The female gametophyte, megagametophyte, embryo sac, one and the same. But in this question, they are asking the microgametophyte is also known as. Microgametophyte is nothing but the pollen. It is a gamete, micro. I have to microspore production. From the microspore only the pollen grain will produce. Process of formation of microspore from the microspore mother cell is called microsporogenesis. Similarly with the megasporogenesis. Can I explain anther? Now anther, anther contain 4 pollen sacs. The pollen will produce from the anther sacs, pollen sacs. I am just in ASRBNET, there will be most of the questions will be in single line. So I am just given the questions in a single line. I am explaining each and everything. From pollen sac, the anther consists of 4.
Prints have been updated to continue. Review the update.
The anther consists of 4 layers. One is epidermis, and endothecium, and middle layer, and tapetum, which is the innermost layer of the anther. It is that tapetum is the innermost layer which will provide the food to the developing gametes. The nuclear division is unaccompanied by cell in the formation now. I told the development process of endosperm. Here there are three types of endosperm. Nuclear endosperm, nuclear cellular endosperm, halobial endosperm. What is nuclear endosperm? The nuclear endosperm means the formation of the endosperm with the repeated nuclear division, not the cell wall formation. You just go to the name itself telling it is the division of only nuclei, not formation of cell wall. What is cellular endosperm? Cellular endosperm, the formation of endosperm by repeated nuclear division followed by the cell wall formation. After nuclear division, the nuclear endosperm is just like the nuclei will divide. But in cellular endosperm, after each division, there will be formation of cell wall. After each individual, there is a formation of cell wall is called cellular endosperm. Without formation of cell wall is called nuclear endosperm. What is halobial endosperm then? Halobial endosperm is the intermediate between nuclear endosperm and the cellular endosperm. We have to, this is very important question. You people have to note the example for each that. Nuclear endosperm is commonly seen in the wheat, rice, barley, oat. The cellular endosperm is commonly seen in petunia, balsam. Halobial endosperm commonly seen in monocots. And you have to even halobial endosperm. Halobial endosperm, there is a I told it is an intermediate between cellular and nuclear endosperm. Nuclear. Here we already know micropolar region and the chalazial region. The cells which are going cellular endosperm towards the chalazial region. The cells towards the micropolar region will go nuclear endosperm. Will develop nuclear endosperm. This is things. This is a definition of the three types of endosperm. For this question, the answer will be nuclear endosperm. The genetic constitution of endosperm, I already told it is a 3N. What is the genetic constitution of perisperm? It is 2N. For each of the things, you have to know the genetic constitution. For example, for the antipodal, it is antipodal, synergid and excel. These the genetic constitution will be N only. For nucellus, it is it is 2N. For integuments, it is 2N. You have to know the genetic constitution of each part. The pollen is produced from, I already told, pollen is produced from anther. I told that also, ovule is developed into seeds. What is stigma? It is a place where the pollen will land and the stigma will recognize the particular gamete for the pollination. The tissue of the seed that develops from sexual fusion of the polar nuclei of the ovule and the second male sperm cell, that term used as. Anyone will answer this question? I expect you to answer this question. I already mentioned so much of time of the definition of the. Anyone? It is endosperm only. I told that it is nothing but the central nuclei will fuse with the another second sperm nuclei will fuse to form a primary endosperm nuclei, that is called endosperm. How seed coat will develop? Seed coat will develop from the integuments of the embryo sac. The pigment responsible for the seed germination. Here the question is pigment responsible for the seed germination, which it is answer is phytochrome. Phytochrome is the pigment responsible for the seed germination. There are 5 phytochromes are there. Phytochrome A, B, C, D. These are necessary for the seed germination. If they fail, if they are not found, there will be occurrence of dormancy. You know, chlorophyll is the photosynthesis pigment. When both male and female flower occur in the same plant but separate, before going to solve this type of question, I will explain what is self pollination, what is cross pollination, what is often cross pollination. Self pollination is nothing but pollination by the same pollen, and it is nothing also called autogamy. Self pollination also called as autogamy. Cross pollination also called as allogamy. And before answering this question, I will explain the mechanism favors the self pollination and mechanism favor the cross pollination. I will explain the each term. You please note it down, and in later part of the this class, so much of questions will be there. Like self pollination, the favored by first and foremost is bisexuality. What is the meaning of bisexuality? Presence of both male and female flower in the same plant, in the same flower, not in the same plant, it is the same flower, is called as bisexuality. It also called as the bisexual flower, also called as hermaphrodite flowers. And other mechanism, which are the other mechanism will favor the self pollination, that is cleistogamy. Cleistogamy. What is the meaning of cleistogamy? Cleistogamy is the pollination. The flowers open, do not open at all. It will favor the self pollination. Even the pollination will also occur, the flower will not open at all. What is chasmogamy? Chasmogamy nothing but flower will open after the pollination only. And other mechanism, like the position of anther. Like in, for example, in tomato and brinjal, the position of anther, like the style of the female flower and the female and the anther will be same, which will favor the self pollination. And some, and in, for example, in pulses also, what is the flower type in pulse? It is peppolinaceous flower. It is very important. Please note it down. It is a peppolinaceous flower in flower. This peppolinaceous flower, most of the pulses, all the pulses are self pollination because of the peppolinaceous type of the flower, where peppolinaceous type of the flower consists of keel petal, wing petal and standard petal. The keel petal will protect the pollen, like not pollen, it is a female gamete. So it will favor the self-pollination. These are the mechanisms which favor the self-pollination. What are the other mechanisms will favor the cross-pollination? The cross-pollination first and foremost is dicliny. It is presence of unisexual flowers. That is presence of only male flowers or female flower is called dicliny. In dicliny there is a monoecious and dioecious. What is dioecious? Monoecious, and what is monoecious first? Monoecious is presence of both male and female flower in the same plant, in the same flower, in the same in different flower, but in the same plant. Sorry, the presence of both male and female flower in the separate, but in the same plant is monoecious. Presence of both male and female flower in different plant. It is different plant. Dioecious is in different plant. Monoecious, it will be in same plant. Is called monoecious. In monoecious there will be two types. You can able to see presence of both male and female flower in the same inflorescence. Presence of both male and female from the same inflorescence is also favor the cross pollination. For example, in mango and castor, if the presence of both male and female in the separate inflorescence, if present in the separate inflorescence but in the same plant, inflorescence but in the same plant. Can you give example for this? That is maize. In maize, you can see that female gamete, female flower, female inflorescence. In maize, female inflorescence also called as silk. The male inflorescence in maize is called as tassel. This is important. These things you have to know. After this, for example, for the dioecious is papaya. And then which are the other mechanism will favor the cross pollination? You know hercogamy. Hercogamy is the hindrance of self pollination because of presence of some other membrane, that is hyaline membrane, which will prevent the self pollination, promote the cross pollination. Example for the hercogamy is, that is alfalfa. And other like this is hercogamy, and like and I told homogamy in the self pollination. Homogamy means maturation of male and female gametes and in the same time. Same time is homogamy. But heterogamy is maturation of male and female flower at different time, which will promote the cross pollination. In heterogamy there is a two condition, protandry and protogyny. Protandry means maturation of anther before the pistil will mature, is called as protandry. I will take next slide. Protandry. It is anther will mature first. If pistil will mature first, then it is protogyny. Example for this type of mechanism is commonly seen in maize and sugar beet These crop will exhibit protandry. Protogamy will exhibited by bajra, that is perblade. These are the different mechanism will favor the self pollination and the cross pollination. Then after discussing all these things, you can able to answer this question, that when both male and female flower occur in the same plant but separate means it's called as monoecious. I already explained dioecious means presence of both male and female flower in the different plant is dioecious. Dichogamy is maturation of male and female at different time. Hermaphrodite means it is bisexual flower, presence of both male and female in the same. Seed coat consists of, how seed coat will develop? I told embryo development. The fusion of egg cell and the sperm nucleus will develop into embryo. It is about embryo, and endosperm means fusion of egg cell and the center nucleus. How seed coat? Seed coat will directly develop from the integuments. The outer covering of the ovule is integument. It will develop into, after the fertilization process, it will develop into seed coat. That is, seed coat is consist of testa and tegmen. What is testa and tegmen? Testa is outer, it is developed from the outer integument. Tegmen is it developed from the inner integument. So seed coat will consist of both testa and tegmen. Answer is both A and C. The microgametogenesis refers to, I already explained microgametogenesis. It is the production of pollen. Testa and tegmen, just now explained, are formed from the integument. Pollen nuclei will, with the fusion with the sperm nuclei, develop into endosperm. Egg cell will develop into embryo. Antipodal cells, they will, after the fertilization process, they will degenerate. Antipodal cell and synergist cell, these will, after the fertilization, they will, during the process of seed development, they will degenerate. The flowers do not open and self pollination is the rule. Is, I told that it's a cleistogamy. It is not homo, it is homo. Homogamy is maturation at the same time. Autogamy is, it's a self pollination only. Allogamy is cross pollination. Fertilized mature ovule is seed. Fertilized mature ovary is fruit. The alluvial layer of the monocot seed. You have to discuss this thing. I told endosperm. There's a monocot seed. We know that. For example, this is a maize monocot seed. It consists of endosperm. And here is the embryonic axis. That is radical. And that is plumule. And that is endosperm. And the outer covering is alluvial layer. The alluvial layer of monocot seed is, it is viable or non-viable, it is dead or none of the above. It is viable only. The living part in the seed, in the endosperm, is the alluvial layer. This year in the SRF question, they has the very simple question. What is the genetic constitution of alluvial layer? It is 3N. But they has like what that they has to match the following. Match the following. They given the crops here and the 3N constitution of the here in the A column they given the crops like rice they given, wheat they given, and maize they given, and some other crop they given. And in B column there's like a chromosome number given. For example, rice they given like 24 plus 12, 36 they given. For wheat that 42. That they have given the pre and constitution here, but we have to match that. So for matching this, for answering this question, we have to know the genetic constitution of the alveolar layer. It is genetic constitution will be 3N. The flower with both functional male and female is called as, it is bisexual also, hermaphrodite also. The rope pulling is a method of supplementary pollination practiced in. What is supplementary pollination? Supplementary pollination is a method where we increase the pollination by pollination of the seed will lead to increase in the seed yield. By some other mechanism will increase the pollination in the crop, which will in turn lead to the increase in the seed yield. In different crops, supplementary pollination will be some other mechanism. For example, in rice, it will be rope pulling. For supplementary pollination in sunflower is by keeping honeybee, by keeping bees in the field will increase the pollination. One example for secondary dormancy. See, here some mixed questions are there. Dormancy. Before going to explain dormancy, secondary dormancy, I will explain dormancy. What is dormancy? Dormancy is the temporary failure of viable seed to germinate due to unfavorable condition. Is a temporary failure of viable seed. You know what meaning of viable? It is live only. It will germinate, but because of some of the unfavorable condition, the seed is not germinated. If the favorable condition comes, the seed will germinate. This is called a dormancy. And what is quiescence? Quiescence is, it is a non-dormant seed which will not able to germinate because of absence of one or more the factor. Because some favorable conditions are there, but some one or more factors like light, temperature, their absence. So seed will go into like a dormant phase, that is called as quiescence phase. In dormancy there are two types. One is primary dormancy and secondary dormancy. That is category. These are types, not times. These are the categories of dormancy. Primary dormancy is the seed produced from the plant directly will be dormant. Like within seeds directly produced from the plant will be dormant only. But after the seed produced, but due to some after some period of time, it will go into dormant means secondary dormancy. After some going some period, it will end at the secondary dormancy. If the seed produced directly from the plant is in the dormant state, it is called as primary dormancy. An example for secondary dormancy. In secondary dormancy there are three types of dormancy. One is thermodormancy, scotodormancy, and also in one light also is there. Light also done. Photodormancy. Scotodormancy is dormancy. Secondary dormancy induced by the presence secondary dormancy induced by the light requiring seed which is maintained in the dark condition. If the secondary dormancy induced by the light requiring seed maintained in the dark condition is called scotodormancy. If the secondary dormancy induced by the seed by the high temperature is called as secondary dormancy and the thermodormancy. These are the three types here in question. Example for secondary dormancy is thermodormancy and scotodormancy. A nucleus seed in a cereal is produced by nucleus seed. You know there are 4 different classes of seed: nucleus seed, breeder seed, foundation seed, certified seed, and some other also classes there: truthfully labeled seed, registered seed. The nucleus is the highly genetically pure seed. It is the genetic purity of nucleus seed is 100%. Similarly, for breeder seed, genetic purity is 100%. The nucleus seed in cereal is produced by, there are different methods. For example, for the cereal, there is a ear to row method is the method followed for the production of nucleus seed. There are 2 types in nucleus seed: nucleus seed stage 1 and nucleus seed stage 2. The method followed for nucleus seed stage 2 is, we have like for ear, I told for it is for nucleus seed stage is ear to row method. You will take one single ear and will sow in the one row. After that, this from one row you will sow the 5 to 10 plants. From 1 year we can get 5 to 10 plants. After that, the from 5 to 10 plant, the seed obtained will be sown in the row to plot. This is nucleus seed stage 2. This is very important. The method is ear to row for nucleus seed stage 1. The row to plot is for nucleus seed stage 2. Why we will do nucleus seed stage 2? Because of the increase in the production of, for you have to produce more number, produce breeder seed production more. If there is indent for breeder seed production will be more, we will go for the breeder seed nucleus seed stage 2. Jerking is a practice adopted in hybrid seed production of. Jerking in hybrid seed production, they have to follow some pollination control mechanism for favor, like we have to for maintaining the genetic purity. For pollination control mechanism, for different mechanisms are there. For example, in rice, for example in maize, there is a hand emasculation, hand pollination, which is also called as detangling. And some in bajra will go direct for hybrid seed production, we will go with the CGMS system. Here they has jerking is a practice adopted in hybrid seed production. In jerking meaning is, what is the definition of jerking is like the some early maturing lines, like plants will come early, like in there is a female plants, female row and male row. The plants, in the male plants, the synchronization of flowering is must in the hybrid seed production. Both male and female flower has to come at the same time and favor the pollination. But in some cases, some plants in the male row will come early, so which will contaminate the female flower, like a female row, and not contaminate, it will come early. Then there is a lack of pollen for the pollination. So to avoid this, with the early coming plants, we will remove first and will encourage the tillering in the, by removing that plant. I will encourage the tillering by giving additional dose of nitrogen and will match the flowering in the female and the male plant. To synchronization of flowering, the jerking is a practice which is followed in bajra. Jerking is the practice for synchronization of flowering, where the early maturing plants are removed first and Isolation could be modified based on the number of border rows in. Before going to answer this question, I will explain what is isolation distance. Isolation distance is the minimum distance required to avoid the contamination there is a minimum distance you will give to there are three types of isolation. One is space isolation, and is time isolation, and barrier isolation. Space isolation is, I already told, it is a distance. We will give some distance. For example, in the hybrid separation of bajra, we will give the 1000 meter from the other field. For time isolation means to modify the blooming date for the male and female plants for the synchronization. Differential blooming date for modifying the isolation distance is permitted in some crops like maize, where here is some condition that it permitted only in the maize where some conditions are there. Only 5% of the plant will have more 5% 5% of plant will come early, like for male flower show matured, and it's okay. I will discuss. I have forgotten this. I will discuss. And some other than this, I will discuss which are time isolation is not practiced in which condition. For time isolation cannot be practiced in tillering crops and indeterminate growth habit crops. The time isolation cannot be practiced in these two types of crop where the crop having tillering, that is bajra and sorghum, and indeterminate growth habits, that is in red gram and brassica, that is mustard. The isolation condition could be modified based on the number of border rows. That is, particularly in maize, we will modify the isolation distance. For example, for hybrid reproduction of maize, we will require the distance of 400 meter. So to reduce, the farmer will have that much of field to give the isolation distance. So it can be modified by using the border rows, planting more number of border rows to avoid the contamination with the offtake. That particularly this practice will follow in maize. In seed production, mechanical admixture in a seed lot affect the physical purity, genetic purity, keeping the quality of the seed, physiological quality. In the varietal deterioration will be varietal deterioration of the seed will be happened by mechanical admixture, and then by outcrossing, and by mutation, and then random genetic drift. These process will affect the varietal deterioration. Here, the presence of mechanical admixture will affect the. There is the quality seed will be defined as it will be physically pure, genetically pure, and physiologically sound, and it should be free from seed-borne disease. It's the quality seed. So if there is a mechanical admixture, what will affect? It will affect the physical purity or else genetic purity, or it will affect from the disease. So if there is a mechanical admixture there, it will directly affect the physical purity. If the presence of off types is there, it is contaminated by the off types and the pollination by the some other plants which is not required by us, it will affect the genetic purity. If pollination by the off type, you will affect the genetic purity. If there is a mechanical admixture is there, it is affect the physical purity. Pre-chilling treatment to seed is given for the overcoming of dash, pest, dormancy, self-incompatibility, maturity. Pre-chilling for breaking the dormancy, there are some mechanism processes there, like first is scarification, tincturing of the seed coat or rubbing of the seed coat is called as scarification, which scarification is commonly used to break the physical dormancy. And stratification, subjecting the seed to low temperature, or else even in the high temperature, that is for low temperature it is called cold stratification. If it is for subjecting the seed to a high temperature, that is more than 15 degrees Celsius, is called as a warm stratification. Pre-chilling also, like pre-chilling nothing but cold stratification. So for for pre-chilling is given for to break the particularly physiological dormancy. Pre-chilling is a treatment given to the seed to break the physiological dormancy. Which of the following is a monoecious plant? Already told, monoecious is the presence of both male and female flower separately, but in the same plant is called monoecious. Which among the following the monoecious mechanism will be there? It will be both in all of the answer for is all of the above in coconut, cucurbits, amaranthus. I missed some like in endosperm part, particularly some in coconut it is a double endosperm seeds. And mosaic endosperm seen. These things I want to tell, but I missed it. So now I am telling. Mosaic endosperm particularly seen in maize. Diploid endosperm is seen in Enothera. Stony endosperm in areca. This I want to tell while explaining the endosperm development. A into B cross is single cross, double cross, three-way cross, top cross. Here given they want to find the single cross, nothing but they in cross between two inbreeds, that is A and B. Double cross is the cross between two single cross. Three-way cross is a cross between inbreed and one single cross. Top cross is a cross between inbreed and the open pollinated variety. These things you have to know. Other than this, what you have to know, particularly from the seed technology point of view, number of isolation required for the hybrid seed production in each type of crosses, like for single cross, double cross, three-way cross, top cross. For maintain for the hybrid seed production, the single cross in single cross, the number of isolation required will be 3. For double cross, it will be 7. In three-way cross, it will be 5. In top cross, it will be 3. How you can calculate? Like just we will see that is for particular. We have to give For this for A we have to give isolation, for G we have to give isolation, for total cross we have to give isolation. Like that we have to calculate the isolation number of how much number of isolation required for the crosses. A method of breeding most commonly used when the desired variation is required to be induced in vegetatively propagated crop. For creation of variation in any breeding population, we have to go foremost thing. The most possible variation will be come from the mutation breeding only. The characteristic feature of the morphological dormancy is the presence of. Before this explaining this question, I will explain the types of dormancy. There are baskin and baskin has given 5 types of dormancy, which is the modified from they given the modified form of the Nikolova system. Where they given 5 types, that is physiological dormancy, physical dormancy, morphological dormancy, morphophysiological dormancy, and combinational dormancy. These are the 5 types of dormancy given by the Baskin and Baskin, where physiological dormancy is nothing but presence of some inhibitor will inhibit the germination process. Physical dormancy is impermeable seed coat. Morphological dormancy is the embryo is not developed properly. Morphophysiological dormancy is a combination of morphological dormancy and the physiological dormancy. Embryo is not developed properly also, and even the presence of some inhibitor will inhibit the germination. Physical dormancy is also a combination of physical and the physiological dormancy. These are the 5 types of dormancy. Here in the question, the characteristic feature of the morphological dormancy is the presence of, like it is underdeveloped embryo. And answer is underdeveloped. Hard seed coat is the form of physical dormancy. If there is a presence of inhibitor, it is a physiological dormancy. Seed germination is stimulated by the hormone. It is by the gibberellin. If the seed germination is inhibited by the hormone, it will be by the abscisic acid. Occurrence of more than one embryo in seed is also known as, it is called as polyembryony. Who given the polyembryony concept? It was given by Leeuwenhoek. Leeuwenhoek given the concept of polyembryony in which crop? That is in citrus. What is apogamy? Apogamy, you know that in apomix, before going to apogamy, I will give the definition of apomixis. Production of seed, formation of seed without fertilization is called as apomixis. Apomixis, there are 2 types. One is gametophytic apomixis and sporophytic apomixis. Gametophytic apomixis is nothing but the formation of seed directly from the unreduced megaspore mother cell. Directly from the unreduced megaspore mother cell, the formation of seed without fertilization is called gametophytic apomixis. Formation of seed without fertilization from the spore, like it is from it It is directly from the introduced megaspore mother cell is called sporophytic apomixis. It is directly from the nucellus. Like it is sporophytic apomixis like directly from the nucellus and somatic cell. That is, sorry, it is from the somatic cell or nucellus cell. It's called as sporophytic apomixis. It is directly from the unreduced megaspore mother cell is called gametophytic apomixis. In gametophytic apomixis, there are three types. Two types: diplospory and apospory. Diplospory is the production of seed directly from the unreduced megaspore mother cell is called as diplospory. Apospory is production of embryo or seed directly from the somatic tissue is called as apospory. Apogamy is production of seed from the synergid cell or antibodal is called as apogamy. The seed which farmer generally use is called as brittle seed, nucleus seed, foundation seed, certified seed. It is certified seed. Hari sir. So this much question, nearly 30 to 40 question I have discussed with you people. For next further discussion, I will take day after tomorrow. Nearly some I will come with a new question in particular in different top. In this session I discuss most of the question from the seed development biology. And I will discuss some some unit question in the further classes. Thank you. Here in our platform we have given a mock test bundle for the ASRBNET 2025. If you want to learn more from these questions, please enroll our mock test bundle where we will get the most of the question. We have given nearly 10 mock tests in our platform. Thank you.
Continue this in next class.